Welcome to our new video concerning the resting and action potentials and how they can lead to neurotransmitter secretion. This is a follow-up video to our previous video where we explained the structure and the functions of a neuron and how neurons communicate with each other. And in this video we want to go more into detail how the signal transmission is actually conducted. Therefore, we will look into three phases which describe how a signal is actually transmitted or why a signal is transmitted from one neuron to another. And it starts by looking at the membrane charge of the neuron. So we take a closer look to the membrane and we can see that there are key factors involved. So we have, first of all, the membrane charge at the so-called resting potential. So that's the default state, which lies around minus 70 millivolts. Then we have our potassium and the sodium channels, and we have the protein pump, which is basically exchanging sodium ions and the potassium ions by using energy by, in this case, dephosphorylating ATP to ADP. And the question, of course, is how this will lead to a signal transmission. So the signal transmission is basically done by having a signal or an impulse, so in this case an electrical impulse, reaching both channels. Because both of these channels are voltage gated. It means they open up if an electrical signal reaches them and is strong enough to open them up. If that is the case, we will have a so-called depolarization. That means the membrane charge starts to further increase and gets into the positives. And this is done due to the fact that we have the sodium channels, which will open up as a result of that. And they are the first ones to open up. And then the sodium ions, as a result, will follow the gradient and then move inside the intracellular space from the extracellular fluid, therefore increasing the membrane charge. Once this has been done, which is happening in a brief moment, the channel will close again. And then we will take a look at the potassium channel, which will open up right away. And in this case, it is the other way around. So we have the potassium ions diffusing out of the cell into the extracellular fluid. And in this case, what they're doing is they are bringing back the membrane charge, which was due to the sodium influx at plus 40 millivolts. And they're bringing them back again to minus 70, since we have the positive ions that are again moving out of the cell. Now, what is happening in this state is also that these potassium channels stay open a little longer than the sodium channels. And the result of that is that we actually even reach membrane charges more negative than minus 70. And this is then called a hyperpolarization once we reach below minus 70 millivolts. And of course, the question arises how we can get back to the resting potential. And this is then done by the pump, because the pump is then still active around the time, so even when the potassium channel is then closed, and it will exchange potassium and sodium ions. And this exchange will then lead to redistributing the ions in their previous proportions, which means we will move up the minus 80 or minus 90 millivolts back to minus 70 millimoles, and then stabilizing the membrane charge. And once we have accomplished that, the cell or the membrane part and the channels are again ready for another impulse, and then restarting the cycle again. And this same approach can also be visualized in a graph. So we also see that there's a threshold. That means if the membrane charge is not able to go higher than minus 55 millivolts, the channels will not open up. That means every stimulus that lies below that is then initially not strong enough and then will end up as failed initiations. If they do succeed in that, so reach above the threshold, they will open up the channels. That means sodium channel open up, it leads to depolarization. We reach the action potential, that means it's 40 millivolts. And at this point we have the potassium channels open up and the sodium are closing. So we will have a repolarization, so positive ions moving out of the cell. And then at this point they stay open a little longer, which then causes a hyperpolarization. And this state is also called a refractory period. So this is the period in which the cell cannot be excited again, in which the cells in the channels cannot be excited again. And it will then use the sodium and potassium pump to basically redistribute the ions to their previous proportions. Again, coming back to the resting state, and then the cycle can of course repeat again. And now to a short recap. So to remember is that a signal is transmitted from one neuron to another, and this is happening through by causing a depolarization at the membrane. And this depolarization concerns the sodium channel, which will open up, and then sodium ions will flow into the cell, so into the intracellular fluid from the extracellular fluid, therefore increasing the membrane charge up to 40 millivolts. Sodium channels close, but the potassium channels open up, and then they do the opposite, so potassium ions move out of the cell into the extracellular fluid, therefore decreasing the membrane charge back to minus 70 millivolts, and also causing an overshoot. That means they cause an hyperpolarization. And to get back to the resting potential, we will use the sodium and potassium pump, and they will redistribute the ions back to their previous proportions. And that's how we then achieve the resting potential. And then the cycle can of course repeat again. And this is then repeated over and over till we reach, for example, a synapse, which you will also see in the previous video that we have uploaded. And then at the synapse, there we have the different process going with the neurotransmitters. And then the whole process starts again by causing an action potential on another neuron. And this is then basically how signals are transmitted between different neurons and different cells. We thank you very much for your attention. And if you have any questions left, any comments, you can leave them down below or under the video. If you are interested in our course or in our project in general, you can also check us out on Instagram, in which we will shortly also upload more content. We have a LinkedIn page set it up and also an email. If you're looking to cooperate with us or joining our team, feel free to contact us and we can arrange it out. And other than that, I wish you a productive week, and hopefully we will see us again.